Today we're going to be talking about the nervous system. The nervous system is a system which comprises of various structures that function to receive, integrate, and send out electrical signals. Now, the nervous system receives signals concerning the environment, either the internal environment or the external environment. It incorporates this information and then therefore functions to release electrical signals to return the body back to homeostatic state. The nervous system is derived from the neural plate. In the third week of development, there is a trilaminar disc. The trilaminar disc consists of the endoderm, the mesoderm, and the ectoderm. Now, the mesoderm contains a tube called the notochord. Now, in the third week of development, the notochord sends signals to the ectoderm above it, which causes the formation of the neural plate. The neural plate has ectodermal origin. Now, the neural plate is formed and then the neural folds are formed. Progressively, the neural folds join together and create a neural tube. The cells of the neural folds, the top of the cells of the neural folds, form the neural crest cells, and those neural crest cells form the peripheral nervous system, while the neural tube forms the central nervous system. Let's go into the cells of the nervous system. Now, the nervous system comprises of the central nervous system and the peripheral nervous system. The central nervous system is divided into the brain and the spinal cord, while the peripheral nervous system consists of peripheral nerves, peripheral nerve cells, peripheral neuroglia. This is the brain and this is the spinal cord containing peripheral nerves. This is the diagram of the spinal cord, and coming out of the spinal cord are peripheral nerves. The brain and the spinal cord. The brain and the spinal cord both make the central nervous system, while the peripheral nerves and all they contain is what makes up the peripheral nervous system. Now, the central nervous system and the peripheral nervous system compose of two major cell types. Now, these major cell types in total make up the entire nervous system, and these are the neurons and the neuroglia. Let's dive into what neurons are and what neuroglia are. This is a typical neuron, or this is the diagram of a typical neuron. Now, a typical neuron contains three major parts, which are the cell body or the soma, the axon and the dendrites. The cell body or the soma, the axon and the dendrites. Now, let's start with the dendrites. The dendrite is a type of neurite. A neurite is a process that is coming out of the cell body of a neuron. The dendrite functions to receive electrical stimuli, and this electrical stimuli will tell the neuron whether to fire or not via inhibitory presynaptic potential or excitation presynaptic potential. We're not going to go into that in this video, but what you need to know is that via whatever neurotransmitter gets in contact with the dendrite, the neuron can actually be elicited to fire or be inhibited from firing. Now, let's talk about the cell body. So, the cell body of a typical neuron is called a soma, a perikaryon, or just the cell body, and it contains major organelles. Remember from our very first video, a cell contains a particular set of organelles, and these organelles aid in the functioning and sustenance of that cell. The nerve cell or the neuron is not any different. It just has some specializations. So now, the major organelle in the neuron is the nucleus. And then the nucleus contains a nucleolus. Now, as you can see here, the nucleolus is drawn in blue, and it is very, very dense. The nucleolus of the nucleus of the neuron is very, very dense. It's very dense because it has a high function in protein synthesis. Protein synthesis is actually very important in the neuron for the synthesis of neurotransmitters through which neurons actually communicate. Another organelle in the soma is the nisal bodies. Now, the nisal bodies are The nuclear bodies are a combination of the endoplasmic reticulum and some polyribosomes. Now, endoplasmic reticulum, if you remember from the very first video, functions in protein synthesis. Remember I said the neuron relies a lot on protein synthesis, both for axonal growth or both for neural growth via stimulation of protein synthesis for microtubules and also for the production of neurotransmitters through which impulse is actually sent or transmitted. Now, the nuclear bodies contain endoplasmic reticulum, and then the nuclear bodies are basophilic, meaning that they stain with basic dye. Okay. Now, let's go. Okay, here. The cytoplasm of a neuron is called the neuroplasm. The cell membrane of the neuron or the cell body is called a neurolemma, while the cell membrane of the axon is called axolemma, and cytoplasm is called axoplasm. Okay, now this highlighted here is a lysosome. I told you we usually have different cell organelles, and then a lysosome, if you remember, functions to engulf bacteria or microbes, whatever is in the cell that isn't supposed to be there, and then forms lipofuscin. Now, a lot of lipofuscin is a pigment. It's an aggregation of different lysosomal bodies. It occurs majorly in aging neurons, because the older neurons get, the more they need to phagocytose elements that are not supposed to be found in the cell membrane. Now, this here is called the axon hillock. The axon hillock is the point at which the soma begins to form the axon. That's just it. And then this here is called the initial segment. Now, the reason the axon hillock, the reason the axon hillock is very important is that, number one, the axon hillock lacks nuclear bodies, but it contains a high amount of voltage-gated channels. This is important because once the dendrites are stimulated and the axon is actually supposed to transmit electrical information, it needs to do this transmission, or this transmission occurs via the influx of various ions. Now, the channels through which those ions can be infused into the axon are majorly located or are very concentrated at the axon hillock. And the initial segment is important because in situations where a neuron needs to be inhibited, it needs to be inhibited from generating an action potential. Other axons will synapse, other axons that already have this inhibitory potential will synapse here. It's called an axonic synapse. Now they will synapse at the initial segment, and through that they can block action potential. They can block the amount of ions that are being infused, and therefore reduce the ability of the neuron to generate an action potential at that particular time. Lastly, concerning the soma, it contains Golgi apparatus. Now, I hope you all remember what Golgi apparatus is for. The Golgi apparatus forms vesicles, empty vesicles, through which various substances can be transported within the cytoplasm of a cell. Now, Golgi apparatus is very important because once protein synthesis occurs, the neurotransmitters need to be stored in the vesicle, and that vesicle is provided by the Golgi apparatus. Now, let's move to the axon of the nerve cell. Now, the axon is also a type of neurite. Remember, not are neurites. Neurites are processes that come out of the cell membrane. Now, the axon is also a type of neurite. It's just that the axon is longer, and it functions in the generation of an action potential, like it generates an action potential. The axon is longer than the dendrites, and the axon functions in the generation of an action potential. Now, these lines, this dash, it wasn't just for me to enhance my drawing. They are actually very important now. They are called neurofibrils. Now, the neurofibrils are part of the cytoskeleton. The neurofibrils are part of the cytoskeleton of the nerve cell, and they just function to give it that structure, that rigid structure, so that the axon is not just flying up and down, flying about. Now, important to note, the axon does not contain any nuclear bodies. The axon does not contain inside bodies. The cytoplasm of the cell or the soma contains inside bodies. Even the dendrites contain inside bodies, but the axon does not contain inside bodies. Instead, or more importantly, what the axon contains are mitochondria, because a lot of energy is going to be used in the transmission of electrical signal. So this is mitochondria. Now, apart from the transmission of electrical signal, some materials are actually transported along the axon, and they can be transported via a retrograde manner or an anterograde manner. The anterograde form of transport is the transport through this direction. It goes through the anterograde form of transport is a transport in which materials go from the cell body towards the telodendron, e.g. action potential, while the retrograde transport is transport through which materials go from the telodendron to the cell body. Lastly, we're going to talk about this. This is telodendron. It is the end of the axon, and then it also is broken down into different dendrites. But this is called the synaptic terminal bulb, whatever you want to call it. But this part is very important because it is the beginning of a synapse. Now, a synapse is the space between two neurons. Now, this is the end of one axon, and this is the dendrite. This is the end of axon of neuron A, and this is dendrite of neuron B. The space between them, because neurons don't touch, is called a synapse. Now, these points are the synaptic terminal or the terminal bulb, and this is where neurotransmitters are released. And then this point contains the receptors through which the neurotransmitters will bind to. That is their functional importance. Now let us go to the classification of different types of neurons. Now I'm going to talk about the classification of neurons. Now, neurons can be classified structurally and functionally. Functionally, they can be classified as sensory neurons, effector neurons, and interneurons. Sensory neurons are neurons that collect information from the environment, whether the external environment or the internal environment, and they send whatever information, they transmit it to the central nervous system, meaning sensory neurons are peripheral neurons. Now, the effector neurons are neurons that send information from the central nervous system back to an organ, a muscle, a gland, whatever. That means that motor neurons are also peripheral neurons, but the central nervous system is the integration, is where integration happens of whatever information. Now, interneurons are the neurons that connect the sensory neurons with the effector neurons. That is the functional classification of a neuron. Now, let's talk about the structural classification of neurons. Now, we have about 4 different types of neurons. We have the multipolar neurons, we have the unipolar or pseudo-unipolar neurons, we have the bipolar neurons, and we have the anaxonal neurons. Now, let's start with the unipolar or pseudo-unipolar neurons. Pseudo-unipolar neurons are neurons that actually have just one process, but that process branches into 2. Coming out of the soma itself, we can see just one process, but that process branches into 2. That is why it is called pseudo-unipolar or unipolar neurons. Now, the 2 processes it branches into are dendrites and attempted axons, but these processes which it branches into are not actively or are unable to be actively differentiated between which is a dendrite and which is an axon. This type of neuron, these pseudo-unipolar neurons, are majorly found in the dorsal root ganglia. Pseudo-unipolar or unipolar. Majorly found in the dorsal root ganglia. Again, this is their cell body, the process, the dendrites, telodendrites sticking out of it. Now, the next classification of neuron is the bipolar neuron. Now, the bipolar neuron has 2 processes, but the 2 processes are one axon and one dendrite. Again, one axon and one dendrite. This is the cell body, this is the axon, and this is the dendrite. Bipolar neurons. Now, bipolar neurons are majorly found in sensory organs like the olfactory epithelium and then the bipolar cells of the eye. The next neuron type we're going to talk about is the multipolar neuron. Now, the multipolar neuron is what I drew here, The multipolar neuron has one axon and multiple dendrites. Again, one axon and multiple dendrites. So pseudounipolar has one process, but that process branches into two. The bipolar neuron has one axon and one dendrite, and the multipolar neuron has one axon and multiple dendrites. Okay, these things branch. This is a dendrite. This is a dendrite. This is a dendrite. This is a dendrite. But it's just this one that is a dendrite. I hope we understand. Now, the anaxonal neuron. My siblings are very, very smart. We already know that means it has no axon. But in the cases, in some sources may say anaxonal neurons do have axons, but they are just very small, undifferentiated from dendrites. But me, I'm telling you that anaxonal neurons in the name, they don't have axons. They just have dendrites, multiple dendrites coming out of them. That is all about the structural and functional classification of neurons. Next thing we're going to go into is neuroglia. The types of neuroglia, their location, either in the central nervous system, the peripheral nervous system, and their various functions. A quick recap: the neuron, the major function of the neuron is for transmission of electrical signal. Whether it be in the peripheral nervous system or the central nervous system, the main function is transmission of electrical signal, and it does this majorly via neurotransmitters. That's chemical signaling. Neurotransmitters are released from the synapse. Now, those neurotransmitters are proteins, and if they are proteins, that means protein synthesis needs to occur in the neuron, which is why our nucleolus the nucleolus of a neuron is very, very dense, very compact, which shows there is high protein synthesis activity. That's also why we have so many nuclear bodies. Nuclear bodies contain endoplasmic reticulum. Let us go to neuroglia.